Okay, so this is the next part, the next new information about the immune system. It is part 2 about the adaptive immune system. Specifically, today we're going to be talking about the cellular component of the adaptive immune system. Okay, so just to kind of put this into perspective, in our last lesson together, we talked about the innate defenses. We talked about that first line of defense, the surface barriers, which we've also discussed in the digestive and respiratory systems as well, because they're just so important in those systems. Also, we have the second line of defense. If something does cross over that skin or mucosal barrier, we can throw all of these things at the invaders. These responses are, kind of like hitting them with a sledgehammer. We don't care what you are, you'll look weird, and we want to get rid of you. If we see you again in a year, we're going to have the same exact response, right? We don't care what you look like, the response is going to be the same. And so all of those are innate defenses. You're kind of born with these defenses, and they don't really develop over time. What you have is what you will always have. What we can change, as the name suggests, are the adaptive defenses. And so the adaptive defenses can change over time. We can train them to recognize specific things. And so all of these defenses protect against specific infectious agents, right? So they recognize this particular bacteria as opposed to that particular bacteria. They have seen it before, and therefore we are going to attack this and not something that looks different. Also, these defenses are going to provide immunity beyond the initial infection site. So all of these innate defenses up here, these have been protecting at the scene of your paper cut. The adaptive defenses are going to pretty much launch a response throughout your entire body. All right, so if a bacteria that gets in through your paper cut is now in your blood, we want to make sure that the rest of the body's tissues are protected against any of the negative effects of that bacteria. And finally, as I mentioned, the adaptive defenses have immunological memory, right? They remember, they know what the surface of bacteria, of fungi, of viruses look like. And the next time the body is exposed to those same substances, they can launch a faster and stronger response against them. And so maybe at this point you start to think about where vaccines come in and why, in kindergarten, everyone's supposed to get the chicken pox or get a vaccine against the chicken pox, right? So, the next time you're exposed to it, you don't get it, you don't get shingles. And so we're going to get into all of those different things. Vaccines are triggering an adaptive response, right? A completely natural adaptive response by your own body in order to protect you against the real thing later on. Right, so that is actually more of something that we're going to discuss in the next lesson together. The adaptive immune system is broken into 2 different parts as well. We had the humoral immunity, which is our next lesson together, or the focus of the next lesson together. Humoral immunity involves the B lymphocytes, or the B cells. These are the ones that make antibodies. And the reason it's called humoral immunity is because hundreds of years ago, we classified different bodily fluids as the 4 humors. And so individuals noticed that there was some kind of recognition, some kind of immunological function of these different fluids. Right, so your plasma, of course, has an immunological function in that it contains a lot of antibodies, and so do the other fluids of your body. And so if, that's where humoral immunity comes from. Cellular immunity is more of what we're going to be focusing on today. Cellular immunity involves the T lymphocytes, and there's actually a lot of different T cells. We are going to be focusing on the helper T cells and the cytotoxic T cells. But of course, there are a few other types as well. And as we go through this, you'll see that this is a fantastically complicated process. I have tried to dial it back for you as much as physically possible. It does vary from what your textbook will tell you. In some cases, there's a little bit more detail. In some cases, there's a little bit less detail. As always, you'll be tested on what I tell you. But of course, I encourage you to read the textbook as a supplement so that you can kind of fill in the gaps or hear content a slightly different way, so that hopefully one of those ways will actually click. Also, I have posted for you on Blackboard a lot of supplemental videos because I know that this can get pretty clunky. And I hope that some of these videos or combination of these videos will help you to understand. Okay, so the adaptive immune system requires cells that ultimately differentiate from those same hematopoietic stem cells in the bone marrow. And so let me draw your attention to this diagram down here. Remember that we have these stem cells that can ultimately detect different types of hormones, different ligands, such as EPO or TPO, that will ultimately drive the stem cells to differentiate into different types of cells. Okay. We, of course, have talked about the platelets, and we've talked about the red blood cells at length, but now, of course, let's talk about the white blood cells. All right, so this is we'll talk about that later part. Okay. Ultimately, all of these different endpoints here, all of these different types of cells have some kind of function in our immune system. The ones that we are going to be focusing on today are these ones, the T cells and the B cells. And we can see that there are lots of different types of T and B cells, which are like subclasses of those initial cells. And we know from previous discussions that hematopoietic stem cells live in the bone marrow. Okay. They are triggered to differentiate into different types of cells. They proliferate, so that means that they undergo many mitotic events. They divide and divide and divide, making more and more of themselves. Those daughter cells can be taken into the blood circulation via those sinusoid capillaries. Right? So it all builds on itself. It's all hopefully being brought together now. Okay. The B lymphocytes, again, this is something we're going to dive into more in our next lesson together, but for now, let me just tell you that the B lymphocytes, or the B cells, are classified as B cells because they express antibodies on their membranes. Right? So antibodies are proteins. They are genetically determined, of course, and so these are really specific to recognizing different types of antigens or different types of epitopes. Okay, so we have a series of different B cells, each one expressing different antibodies on the surface of their cells. And so that's actually what is being shown down here. Each one of these little Y-looking fellas is a type of antibody, and so it's going to recognize a particular antigen. Okay. The B lymphocytes are going to be made entirely within the bone marrow. So that's what this image is showing us up here. Here is a stem cell. Here is the B cell that has been produced via mitosis or proliferation. And it's going to grow up entirely within the bone marrow. So it's going to be trained to recognize only that specific type of antigen. It's going to mature and therefore be able to recognize the antigen versus self, right? So our own cells, we don't want to attack our own cells, only the stuff that is foreign. Okay? And so once the B cells have grown up, right, they are going to be taken into the blood circulation via the sinusoid capillaries in the bone marrow. And so we talked about a while ago. These B cells have not yet been exposed to that which they can recognize, right? So even though these little antibodies down here can recognize one particular type of antigen, they haven't actually seen that antigen just yet. And so the B cells with their antibodies are going to be washed away in the blood, right? They are ultimately going to leave the blood, be taken into the lymph fluid, right? Or the lymph fluid is going to be taken into the lymphatic vessels, and from the lymphatic vessels, the B cells are going to populate. They're going to hang out for a little while in assorted lymphatic structures. Okay, and so what we now call naive B cells, because again, the B cells have not actually seen the antigens that they're supposed to recognize. They are still naive. They still haven't had that first encounter just yet. And so they are going to hang out in the lymph nodes, pretty much patrolling the lymph as it flows through the nodes, or it's kind of like milked through the nodes, looking for that particular substance. Okay, does that make sense? Are there any questions about that? To be continued for sure. All right, and so I feel like this concept really is hard to visualize, and so I want to kind of rewind just a tiny bit, remind you how this works. So again, the B lymphocytes are going to be made in the bone marrow. The bone marrow is within the spongy bone of the long bones, of the sternum, of the pelvis, etc. And so once those B cells are created, they are going to enter into the blood circulation, right? So they're going to enter in these capillaries, and they're going to be circulated around in the body. And ultimately, in a tissue, so in the legs or in the arms or, in the head, wherever, these B cells and T cells, as we're going to get into, are going to actually leave the blood vessels. So here is a capillary bed. And so here is a lymphocyte. Specifically, it's a T cell, but B cells do the same thing. They are going to squeeze in between those endothelial cells of the capillaries of that tunica intima. They're going to squeeze out and ultimately enter into the tissues, right? The leg tissues. Okay. We know that once cells are out of the circulation, the blood circulation, they can be sucked into these blind-ended, super porous lymphatic capillaries. And the lymphatic capillaries are, of course, woven into the systemic blood capillaries. And so, again, they are washed into these lymphatic vessels and ultimately milked along, passing through many lymph nodes along the way. And so they get to hang out in the lymph nodes. And while they're in this particular lymph node, more fluid is being milked up from, right, the leg tissues, wherever. And so the B cells actually have a front row seat to anything that is hanging out in our tissues, anything else that got washed into these vessels. All right, so if there's anything in the legs or anything downstream of where this B cell is going to be hanging out, it is going to see it as that fluid flows past. And frankly, if this B cell doesn't see its particular type of pathogen, maybe a B cell in the next lymph node will, or the next, etc. Does that make sense? Again, I feel like one of the hardest parts of this is just kind of visualizing where all these different things are. So I'm going to try to step back and show you where these events are occurring as much as possible. Okay. The T lymphocytes. Okay. Remember that the T lymphocytes are also descendants of those same stem cells. Okay, so the same stem cells in the bone marrow. In the bone marrow, the way that the T cells actually become T cells, right, as opposed to B cells, is that they have different types of proteins expressed on their membranes. Okay, so B cells, of course, have these antibodies expressed on the surface. T cells have several types of membrane proteins. First of all, we have a series of cell differentiation glycoproteins. All right, so the glyco is just telling us that there's a little bit of sugar on our proteins embedded in the membrane, and cell differentiation is pretty much telling us that the identity of the T cell, right, as a T helper cell or a cytotoxic T cell, is determined by these types of proteins. Right, so what we can see here is a T cell. There are different types of glycoproteins on the surface of the T cells. Ultimately, the type of glycoprotein, either a CD4 or a CD8, is going to determine the name of this particular T cell. The two main ones that we're going to talk about are the T helper cells and the cytotoxic T cells. So T helper are going to kind of help different immune responses, and cytotoxic are actually toxic to cells, right? So they're actually going to be killing different invaders, right? And I go like this because they're like poking holes in them and allowing all their guts to leak out, and therefore they die. Okay, so again, the surface proteins that determine the identity of the T cells are CD4 for T helper cells and CD8 for cytotoxic T cells. Okay. The T cells also have T cell antigen receptors, which we can abbreviate as TCRs, T cell receptors. These T cell receptors are responsible for actually seeing the antigens, right? So where we're going with this is that part of the innate immune system involves antigen-presenting cells engulfing or phagocytosing an invader, chopping it up, and putting pieces of that invader on the surface of the cell. Right? And so they do that in MHCs, right? Major histocompatibility complexes, MHCs. And so here is a piece of that invading bacteria, for example, and here is the MHC. And so the importance of this is that the CD4 or CD8 glycoproteins recognize the MHC, right? So this protein here is essentially saying, oh, like we know this, that MHC looks like us, so this APC is good, right? We're not going to attack that. We recognize what is happening here. The MHC is going to present a piece of this invader. And so it is the T cell receptors, the TCRs, that are actually going to look at this particular antigen and essentially launch an immune response against that antigen, right? And so we need to, of course, as always, recognize self, right, with the T cell or with these glycoproteins, and we need to recognize non-self here with the T cell receptors. Okay. Note that it is the glycoprotein that is recognizing the MHC, and so each glycoprotein, CD4 or CD8, can recognize only one type of MHC. Okay, so CD8 recognizes MHC1, CD4 or the T helper cells recognize MHC2, which are the much more common ones. Okay, does that make sense so far? Okay. Normally, I would draw all this crazy stuff out for you on the board, and we kind of go back and forth with the PowerPoint. I'm going to do that today to the best of my ability, and so I'm going to kind of share and then stop sharing and everything as much as I can here. And so ultimately, I am going to be walking you through, or I'm going to be drawing for you a diagram that's going to end up looking something like this, so big and clunky and complicated. In my next lesson with you guys, I will scan this diagram in and actually point out the little details here. But for today, I think it's really important to draw these things together so that we can physically feel the flow of the events and from different places and see exactly what is happening here. Yeah, bear with me. I try to use a Blackboard super old school style, okay? Okay, so I have already drawn the hematopoietic stem cell up here, right? So this is within the bone marrow, okay? Remember that there are triggers, there are ligands that are going to cause the differentiation and proliferation of these stem cells. One option are the B cells. All right, so these stem cells can grow up to be the B cells. Okay, and remember that the B cells contain antibodies on the surface of their cells. Right, so abbreviated ABS, antibodies. Okay, the other thing, and of course what we're focusing on a lot more now, are the T cells. Okay, So at this point, we know that the T cells have a series of proteins on their surface. Okay, all of the T cells are going to have green. Hopefully you guys can see the different colors here, but T cell receptors. Right, they're going to be important for recognizing the pathogen itself or the antigen itself. We also have at this point in the bone marrow, we had the addition of both of the different types of glycoproteins. So here we have in red a CD4, and in purple we have a CD8. Okay, so in the bone marrow, we're actually adding all of these different proteins. These are not yet mature. They are not yet able to recognize particular things. In the bone marrow, we pretty much just throw all of these different proteins on at the same time, and we'll kind of sort it out later. Hey, does that make sense so far? Is that okay? Okay, so these particular cells, right? So these T cells that can't really differentiate between things just yet are going to go to an organ called the thymus. Okay, so they're going to the thymus, right? So all of these proteins are added on the cells within the bone marrow, and then these cells themselves are going to go to the thymus. Okay, is that good so far? Okay. Or if not, you will tell me. All right, so the immature T cells, these guys here, are going to, again, be taken into the sinusoid capillaries. They are going to circulate around in the blood. They're going to exit the blood. They're going to be taken into the lymph, right? Be taken ultimately, and it takes a little bit, into the thymus. Okay, again, these are not yet differentiated into CD4 or CD8. They have everything at this point. Okay, so what exactly is happening in the thymus? Well, the T cells are called T cells because they are they are the thymus cells. They are trained in the thymus. And so what exactly does this training involve? Well, it involves teaching these T cells to become immunocompetent and self-tolerant. And so what exactly does that mean? Well, here we can see some cells that are within the thymus, right? So these are like the tutors in the thymus itself. And these are the T cells, right? So they're developing, they're still really immature. They still don't really know what's going on quite yet. And so we can't just like release them willy-nilly out into the body because they might attack the wrong thing. And so these little tutoring thymic cells are going to teach the T cells to recognize self-MHC, right? So recognize MHC class 1 or MHC class 2, and don't attack it, right? This is us. This particular shape of a molecule means that this molecule belongs to one of our cells. We really like our cells and we want them to stay where they are. And so we cannot attack those cells. Okay. Again, remember, this is something from the last lesson that I gave you guys. This is an MHC. We can see part of the protein within the membrane of an APC. This here is the component of the MHC that actually binds to the piece of bacteria or whatever pathogen. And this component down here is ultimately what the T cells are learning to recognize, right? This looks like us. Don't attack anything with this particular shape on it. Okay. Also, during this time, the T cells further differentiate. That is, they are going to grow up here to become a cytotoxic T cell, right? So T sub C, or a T helper 0 cell, right? And so as we're going to see, there are a couple different types of T helper cells. At this point, the T helper cells haven't yet determined which of the two T helper cells they're going to be, but they are indeed T helper cells. They cannot rewind the clock and become cytotoxic T cells. They cannot rewind even farther and become a B cell. They are committed at this point to being either a T cell or a cytotoxic or a helper T cell. Okay. And so the way that happens is that essentially one of the types of glycoproteins are removed from the surface. So either these cells remain with the CD8 or they remain with the CD4. Okay. And so ultimately, after this process happens, right, after this training, right, we know what looks like us, we know what doesn't look like us, and we can actually recognize and attack, or at least produce a response against stuff that doesn't belong, right? And so these cells are now called naive cytotoxic T cells and T helper 0 cells, and they are going to migrate to the secondary lymphoid organs. Again, going back to this here, all of that training is happening here in the thymus. Okay. And from the thymus, once the T cells are good to go, essentially they are going to jump into the lymph, migrate all around the body, and ultimately populate these different lymph nodes. Right. Again, so they have a front row seat to anything that is floating around in the fluids of the body, making sure that all of those things are good to go. Right. They belong to us. They are expressing the appropriate MHCs. All right, questions? Okay, so let's draw this in up here. Okay, a little bit of back and forth today. Okay, so these guys are in the thymus being trained. Okay, what I hadn't mentioned before was that this training process is super rigorous. That is, most cells fail. And so most of these cells are going to fail. In fact, 98% of them are going to be killed off, right? We really don't want cells that can't recognize us versus not us. We don't want to release them into the body. If our cells are a little bit too excited, or rather, sorry, if these cells were to go out into the body and they couldn't differentiate between self and non-self, what that would mean is that, so if they fail in a positive way, or sorry, if they fail in a negative way, they are essentially unable to recognize self versus non-self, and so this can lead them to attacking our own cells, and so that would produce an autoimmune disease. Okay, so these are overzealous. They cannot recognize self versus non-self. If they fail in another way, so it's called the positive way, essentially, they cannot actually launch an immune response. They are not immunocompetent. And so if we release those types of cells out into the body, that would essentially produce an ineffective immune response. Okay. All right, so we don't want to attack ourselves. We don't want to be unable to attack things that do come into our body. And so literally the overwhelming majority of our cells die off, right? We kill them. So 98% are destroyed. Okay. The other 2%, what is happening with them? And I'm totally going to run out of space here. I'm really sorry, but we'll figure it out. So the other 2% are going to migrate out to our tissues, right, to our lymphatic tissues. And so these ones have further differentiated. So now these are called either, depending on what they have grown up to be, naive, cytotoxic T cells. Okay, so they're naive because they still haven't seen what they're meant to see. Cytotoxic, why would these be cytotoxic? Well, they are lymphocytes. They are T cells, so they have these T cell receptors right here in green. And they only have the CD8 co-receptors. All right, remember way back here in the thymus, they used to have all of these different things. Now, because they have been trained, because they are now more mature, they got rid of the CD4 and remain with the CD8. Okay. So now we can call these cytotoxic T cells. Okay, so that's one type of cell that we've produced. And again, if I am leaving you guys in the dust or not being clear or you can't see, please let me know. Okay. The other type of cell, right, also migrating to the lymphatic tissues because it has fully differentiated. These are the T helper cells, so T sub H. Okay, just like any other T cell, we're going to have the T cell receptors. Always this kind of triangle guy here with in green. And these cells have lost their CD8 receptors, only keeping their CD4 co-receptors. And again, these are naive cytotoxic, naive, because they haven't seen what they're supposed to see yet, helper T or T helper cells. Okay, another word or other terms, right? There's always more terms for these type of cells. Helper T cells, sorry. So the other words for helper T cells are also CD4 cells. These are also called CD8 cells. Right? So depending on what resource you're looking at, you will see them referred to exclusively by their co-receptors. Right? So these extra glycoproteins. Okay. Makes sense? Is that good? Kind of, to the extent it can be? All right, so these are in the nodes. Okay. Okay. Only 2% of them are surviving and can go to the nodes. Okay, so what's happening in the nodes? Remember from our last lesson together that there are many lymphatic vessels that flow into the convex surface, right? Kind of like the greater curvature portion of the lymph nodes. Lots of this lymph filled with all sorts of different things coming into the node. The node itself has this little framework, this architecture within it that the T cells at this point, and as we're going to learn, the B cells can latch onto and patrol the fluid that's flowing through. The lymph fluid is going to kind of hang out in the node for just a little bit. All right, remember that there's only one exit point and multiple entry points, and so just like merging lanes of traffic, there's going to be a little bit of a traffic jam. You're going to sit there for a little while so that everyone can merge into one lane. And so here, this allows the lymph to be monitored by these cells a lot more. And so remember that these T cell receptors are proteins. They are produced by a genetic code. Proteins bind specifically and tightly to their substrate. And so these guys are looking for something specific, okay? Specifically something that's presented on different MHCs, okay? All right, so the lymphocytes hanging out in the nodes here are going to recognize antigens, particularly those that have been presented on MHCs, right? So MHC class 1 or class 2, depending on which cell we're talking about, we're getting there. And they're also going to recognize antigens that they've seen before. Okay, so before we continue with this, just want to take a step back and do another poll. All right, I think you guys can see that now. So B cells are trained where? While the T cells complete this training process, right? So they kind of grow up and they learn self versus non-self. Where? All right. So this should be on your screen. I know that not everybody or not every device can actually participate in the polls, and I'm really sorry for that, but hopefully even if you can't click on the answer that you think, hopefully you're at least thinking about the different answers, thinking about why are B cells B, why are T cells T? I'll wrap this up in just a couple seconds here. It seems like the majority is on the right track, right? And that B and T is really a clue there. All right, and so you guys did it again. Awesome. B cells are B because they're trained in the B for bone marrow, right? They are T cells because they complete this training in the T for thymus. Okay, so again, they're all born, they're all made in the same place, right? They all start with those stem cells all the way in the bone marrow, and then the B cells grow up in the bone marrow. The T cells, get shipped off to their aunt to be raised, and then they can go forth and do wonderful things with their lives. All right, so the correct answer here is C. Questions? All right, one more. I'll clear this out. Okay, so in the thymus, what exactly is this training? Right, in the thymus, the T lymphocytes learn to recognize self versus non-self. Are they presented with specific antigens so they can recognize that shape later? Right, so this is bacteria A, this is bacteria B. Do they differentiate into cytotoxic or helper T cells as opposed to having both? Are all of these true, or only A and C? Think about what the word naive means in this context. Maybe not as a cultural context. All right. So about half of you guys have answered. So I'm going to end the polling and share the results. And this is a little tricky. That's why I'm giving you guys these questions now, so that you don't kind of learn the wrong things and study the wrong things. The correct answer is actually E. And so there was really close between D and E. So why is B false, essentially? In the thymus, we are not actually presenting, excuse me, oh my gosh, so much chalk. Chalk. Anyway, the T cells are not actually presented with, here is a bacteria, here is a virus. None of that stuff is happening within the thymus. The thymus is merely teaching the T lymphocytes to recognize, this is the self part of the MHC. We recognize this, don't kill that. versus here's the thing that's presented on the MHC. That doesn't look familiar. So we should recognize whatever is actually being presented on the MHC. Okay, so the answer again is E. All right, so let's talk about the actual presentation part. Right, so these guys are chilling in the lymph nodes. Right, and so how do these T cells, cytotoxic or helper T cells, how are they actually exposed to the pathogens? All right, so let's rewind just a little bit to what we talked about in the innate system, because this is the crossover point. We know that at the scene of damage to the surface barriers, invaders, so in this case we'll talk about a bacteria, can get into the skin. We know that neutrophils just kind of eliminate one of these and then die. The next one eliminates the next bacteria and dies, etc. Excuse me. Wow, it's going to be a problem. Mast cells are going to release chemicals such as histamine, which increases the inflammatory response. Okay, we can see that those chemicals are going to increase the permeability of the blood vessels. They are going to lead the area to become red and hot. And also they are going to call over more of these cells. Okay, these front end fighters here. Also what's happening, and this is really where we get into the adaptive phase, macrophages or other antigen presenting cells are going to engulf or phagocytose the bacteria. Okay, this leads them to do a couple things. First of all, it releases more cytokines. The macrophages can also release chemicals to induce the healing process, right, of this wound here. We want to fix that. And also, just a throwback here, here are the platelets, which are forming that platelet plug and hopefully stopping the bleeding, hopefully stopping the invaders from coming in. Right, so this is what we're focusing on now. The macrophage is just engulfed or phagocytosed in invading bacteria. Okay, what is it going to do? All right, the same things that we talked about in the last lesson. APC, antigen presenting cell, is going to chop up that protein or that pathogen. Okay, so here, let's see, phagocytosis. We're going to chop it up and actually attach it to MHCs. Right, so here are the MHCs that are made, they're transcribed and translated, and here we're going to stick them onto those pathogens. The pathogens are now dead, they can't hurt us anymore, but we want the T cells to know, hey, this is what's coming in, and it's possible that these bacteria are going to get into the blood. And if that happens, we really don't want the bacteria to be shipped off to our digestive tract and our muscles and all these other places. We want to get rid of it. We want to launch a whole body response against what? Against this. All right, so this is kind of like the show and tell mechanism here. The macrophage is going to, let's see, it's going to stick those MHCs with their little antigens on the surface of the cell. Right, and while all these different things are happening, the antigen presenting cells, in this case the macrophage, is going to be taken into the lymphatic vessel, right? And it's going to be milked along those vessels and ultimately pass through one of those lymph nodes. Okay, in the lymph node, the APC is going to present the antigen on the surface of its MHC to a T cell, right? And remember that the T cell receptor, this little blue line here, is going to recognize that, this MHC looks like us. This is good to go. This must be show and tell, not let's get rid of it. And the co-receptor, recognizes that this MHC is good to go. This is us. And the T cell receptor is going to look at this little antigen and say, we need to launch a response against this thing in particular. Okay. And so the fact that we have two different classes of T cells that we're talking about, the helper T and the cytotoxic T, this should imply to us that there are two different types of antigen, or minimally two different types of MHCs that the two cells are going to recognize. And that's absolutely the case. MHC class 1 is great at binding to funky cells, right? So any cells that are infected are going to be human cells, that is, that are infected with something are going to be placed on MHC class 1s. And I mentioned viruses here. If you haven't taken micro, or even if you have, just a reminder, viruses are not technically alive. They can't actually they can't reproduce on their own. They can't make their own proteins or their own RNA or anything. And so they actually inject their genetic material into a human cell, and the human cells stop doing what they're doing, right? I'm going to stop being a neuron, and all of a sudden I'm going to start making more virus proteins. I'm going to make more viruses. And then eventually the cell, right, our human cells, are going to look very different, because no longer are they maintaining themselves, no longer are they completing the normal function. Now they are morphing into this essentially virus factory. Okay? And so MHC class 1s are great at gripping onto, at presenting human cells that have been infected by something like a virus, right? So that's why viruses are so well, one of the many reasons why viruses are so difficult to treat. First of all, they're inside our cells, right? And our cells are making more of them. So in order to kill them, right, we're kind of killing our own cells, and that's obviously not ideal. Also, viruses aren't alive, right? Normal antibiotics impact the living components of cells. And so viruses aren't actually alive. And so normally antibiotics aren't going to work on viruses. Just a little bit of a side note, very relevant now, right? What I'm trying to get at here is that MHC class 1s are great at presenting infected cells. Usually this is associated with something like a virus. MHC class 2 are more universal. They can present either intra or extracellular pathogens, right? So just the normal old bacteria that's floating around, or they can present infected cells as well. And again, I just want to remind you that the conserved portion down here, the ones that those co-receptors are actually recognizing, this tells those T cells that this is us, don't attack us. But there are two different configurations. As we can see, there's no connection here. And so this is a class 1 molecule, and this is a class 2 MHC molecule. Okay. All right, so questions about that? All right. All right, so as I said, the APCs, once they have phagocytosed whatever the invader is, whatever the weird looking infected cell is, they are going to be propelled along those lymphatic vessels to the lymph nodes. And there they are going to present whatever they have to the T cells. Okay. So we'll draw in some extra stuff here in just a second. Before we talk about CD8 versus CD4 cells, let me also mention that lymphocytes, these T cells, are going to be activated by the APC saying, hey, look at this, attack this. But not only that, okay? We need something else, right? It's really critical to make sure that our immune system only attacks stuff that we want it to and not stuff that we think is pretty good. And so what we're going to see up here is that an APC presents its antigen, and of course that T cell receptor is going to recognize that antigen and launch a response against it, right? However, we also need a reinforcement. We really do not want to make a mistake here. And so that reinforcement comes in the form of cytokines. Remember that I told you that cytokines are a class of chemicals that is going to regulate the production of white blood cells. And there are lots of different ones. It's not important for us in this class to know the differences between all these different cytokines, But what we do need to understand here is that cytokines are going to be produced by the APC. And if this is a serious threat, if this is for realsies something that we need to go after, cytokines are going to be released at the same time that we get the presentation of the antigen. So the T cell, whether it's cytotoxic or helper T, is going to receive two messages simultaneously. Here's the thing we need to go after. And the cytokines know really, like, this is important. You need to launch a response against this. If there is no cytokine reinforcement, which we technically call co-stimulation, this is going to lead to inactive T cells, right? So there's no response or functional inactivation, which is called anergy. And so if we don't co-stimulate those T cells, we pretty much, our body, our immune system cannot recognize that substance again, right? Because we never got the message that this is a problem. And so anytime we see that in the future, we're not going to respond to it appropriately. Okay, so anergy is, we detected the antigen, but we did not actually activate our T cells in response. Okay. The other thing that this co-stimulation is doing is it can determine the type of response, right? So I told you that these cells differentiate into CD8 or CD4, but there's one extra level. These guys can further differentiate. They can be a T helper 1 cell or a T helper 2 cell. And essentially the difference here is that, or is essentially the different parts of the immune system that are activated, right? Essentially, and I know I'm running out of time here, so I want to summarize so you have some kind of context as I continue later. One type of T helper cell is going to attack intracellular invaders. The other T helper cell is going to lead to the attack of extracellular invaders. And so it is, as you might imagine, a really different response. If we're attacking things that are outside our cells, just in our fluids, versus actually inside of our cells, right? It's two different sets of technology. And it is this here, right? This co-stimulation event that determines what the T helper cells are going to grow up to be, and therefore what they are going to grow up to stimulate, right? What part of the immune system they're going to activate. Okay. Again, any questions there? Okay. All right. So this image kind of summarizes the cytotoxic T cell activation as well as the actions of the mature cytotoxic T cells. I will do my best to draw a little bit of this on whatever is left of the board up here, and then I will continue with this in another video or at another time. This is step number 4, and I say 4A because the process is just a teeny bit different between the cytotoxic T cells and the two types of T helper cells, and for the B cells, which is next class. So what we are seeing here is a macrophage phagocytosing an antigen, chopping that antigen up, presenting it on the surface of its cell in an MHC class 1. Okay. So the cytotoxic T cells, the CD8 co-receptor, is going to recognize specifically MHC class 1. Okay. And so any kind of intracellular invaders, such as a virus or something else that can actually get inside the cells, are presented on class 1. Okay. When that occurs, right, we also need some co-stimulation, right? And once the cell is activated by co-stimulation as well as the presentation, the cytotoxic T cell is actually toxic to cells. It can destroy the particular antigens. All right, so let me one more time stop sharing and add to our image up here. Okay. The APC has just come in from our paper cut or whatever, our lungs, wherever we are, and it is going to present on in MHC class 1. So MHC class 1, some kind of an intracellular pathogen. Here, let's draw this in. Nope, we're going to do this. So here is, we'll say, the virus infected cell, or a piece of it is going to be presented to the naive cytotoxic T cell, which is just hanging out within the lymph node. At the same time, we are going to get co-stimulation. Okay, so co-stimulation by different types of cytokines. Okay, and again, the different types of interleukins, which are a cytokine, are not critically important to our understanding now. Co-stimulation and presentation is going to lead to the creation of more of these cells. All right, so this here is the activation and proliferation. Okay. And so this proliferation is important. We don't want just one cytotoxic T cell to know what the heck is going on. We want to make tons of these so that tons of these can go out and circulate throughout the body and recognize this antigen in particular. Okay, so proliferation is essentially mitosis. Mitosis of what? Of this particular cell that recognizes this particular antigen. And so when you go to the doctor and you say, oh, I'm feeling ill, he usually feels like your lymph nodes up here. And if they're kind of swollen, generally what that indicates is that proliferation is happening. So all of this mitosis is occurring within the lymph nodes. They are going to get swollen and uncomfortable because anytime you have a lot of mitosis happening at the same time, it's going to be a little bit uncomfortable. Your tissues are going to stretch a little bit with the additional cells, right? So that's why he's feeling for your nose, your glands, to make sure, or to figure out that this process is happening in your body at the time. Okay, and so these cytotoxic T cells are now going to be mature. Mature cytotoxic T cells, these are activated, they are ready to go forth and do their job. And so what exactly is their job? Well, of course, they are going to be released into the blood or into the lymph. And so they're going to circulate throughout the body looking for anything that looks something kind of sort of like this. Okay. Cytotoxic T cells specifically are toxic to cyto2 cells, and therefore they can actually kill anything that looks like this. Okay, and so in order to do this, they release something called Granzymes. Okay, we saw this before as part of our innate immune system. Granzymes are released, they poke holes in the cell, and essentially the cells like leak out all their contents, and therefore they die. And, so does that make sense? All right, so let's look at a question before we continue on with our story up here. And I did make a couple changes so that we could fit a little bit more stuff up there. I'll show you that in a moment. What is required to produce abundant cytotoxic T cells or proliferation, which recognize specific antigen A, right? So this particular bacteria. Activation of a stem cell by a B lymphocyte, phagocytosis and presentation of the pathogen by an APC, co-stimulation of a cytotoxic T cell with cytokines. All right, so let's just walk through these three and see if it's just one of these three answers or two of these answers. So activation of a stem cell by a B lymphocyte. Doesn't seem like we talked about any of that, and definitely haven't mentioned any actions of B lymphocytes at this point yet. And so the stem cells themselves are actually activated and triggered to differentiate and proliferate by different types of chemicals like cytokines, EPO, and TPO. So that one seems like it's not quite right. Phagocytosis and presentation of the pathogen by an APC. That one seems a lot right, or a lot closer to being right. So we had the pathogen phagocytosed, and now it's presented by an APC to the cytotoxic T cell here. And so that answer seems to be correct. And C, co-stimulation of the cytotoxic T cell with cytokines. And so that is this right here. So we need both the co-stimulation and the presentation in order to get the activation, right? Like this is now mature, it's turned on, and proliferation, making lots more of this particular cell that recognizes this particular thing. Okay, and then all the descendants of this particular cell are then able to go into the blood, into the lymph, and they can actually kill whatever cells look kind of like that by releasing Granzymes, which pokes holes in those cells and causes their destruction. Okay, so the correct answer here would be E, both B and C. Okay. And finally, why do your nodes or glands become swollen? It's a little bit harder, a little bit puffy when you're fighting a cold. A, the invading virus triggers an inflammatory response in your lymph nodes. B, activated cytotoxic T cells are proliferating in these lymphatic tissues, or C, neutrophils are proliferating at the site of infection, which is in the lymph nodes. So A, the invading virus triggers inflammation. Yes, there would be an inflammatory response, but only really where that virus is located perhaps, and so not specifically in your lymph nodes. You would be having a lot of other inflammatory symptoms in response to the virus, right? Activated cytotoxic T cells are proliferating in these lymphatic tissues, and yes, that is in fact the right answer. The answer is B. And so anytime you get a lot of proliferation, your tissues kind of get puffy and swollen. Their tissues are stretching out to accommodate all those additional cytotoxic T cells. And really anytime you're growing or replacing tissue, undergoing lots and lots of mitosis, there will be a little bit of soreness there. And finally, neutrophils are proliferating at the site of infection. Maybe that is possible to an extent, but the lymph nodes are certainly not the site of an infection. Okay, so I just wanted to go through those questions before we carry on here. And I have made a couple modifications to the diagram since we saw this last. Right, so what I want to show is that in the thymus, all of the T cells are trained, right? And then once they are trained, they are naive cytotoxic or helper T cells. They circulate into the lymph nodes where they are going to populate said lymph nodes. And so in the lymph nodes, we have CD8 cells hanging out looking for anything that doesn't belong. So way over here, the APC. And also, we have T helper 0 cells, right? So these ones still have one additional level of differentiation, and that level of differentiation is only going to occur when and if they are exposed to a particular type of pathogen. And so I have added an extra one in here. That one was already in the last diagram. I just want to show you that there are two different options. These are identical cells, but this guy is going to be exposed to one type of pathogen, that one is going to be exposed to a different type of pathogen. And so let's take a look at these first, and then we'll draw them into the board diagram up here. All right, so T helper cells recognize pathogens that are presented on MHC class 2 proteins, right? So the CD8 cells recognize MHC1, T helper cells recognize MHC2, but MHC2, of course, can present a lot more different types of pathogens than the MHC1. So if a pathogen is intracellular, so it's inside a cell, it has changed something going on inside the cell, it is going to be presented, or when it is presented, it is going to trigger the T helper 0 cells to differentiate into T helper 1 cells. Okay. And so when that happens, essentially the T helper 1 cells are responsible for not directly killing anything, right? They're just helpers. The cytotoxic, the toxic to cells are the only ones that can physically kill pathogens, but the T helper cells are essentially the enablers. They essentially give the gas to different aspects of the immune system to more strongly respond to this particular pathogen. And so the T helper 1 cells are generally going to launch a stronger response against intracellular pathogens. And so more or less, this is going to trigger the innate immune system, right? So it's going to increase the numbers and activities of macrophages and natural killer cells, right? Both of these are part of the innate immune system, and so they are particularly good at dealing with intracellular pathogens. T helper 1 is going to trigger a pro-inflammatory response, right? And it's also going to enhance the activity of cytotoxic T cells. Remember, the cytotoxic T cells are also looking to combat against intracellular pathogens. And so if the T helper 1 cells trigger a stronger response of the cytotoxic T cells, they're both kind of working towards the same goal. We're both trying to get rid of intracellular pathogens in particular. I also, T helper 1 cells are going to activate B cells. That's a detail for the next lesson that we have together. But what I want to mention here is that there are 5 different types of antibodies. Depending on which antibodies the B cells are told to produce, that is going to affect the activity of those antibodies and essentially where those antibodies can work, right? So the location and how they're working. And so IgG antibodies in particular are great for complement, right? So again, this is just another way of enhancing the innate immune response. Right? So the innate immunity is doing its thing, the same way all the time, but T helper 1 cells in particular can really ramp up that response. And so all of those other components produce a stronger response ultimately. Okay, so let's add this into our diagram here. Okay. So here, a T helper 1 cell, if there is an APC, that nucleus, that is going to present on an MHC2, a type of intracellular pathogen. Okay, so this is intracellular antigen. Okay, together with the presentation, we also need co-stimulation, right? We don't want anergy. So co-stimulation. And that interaction, the presentation of an intracellular antigen, as well as the co-stimulation simultaneously, is going to activate and proliferate these particular cells. And so now these cells are your T helper 1 cells. Right, it's just a subtype of T helper cell. Right, and what exactly are they going to do? They are going to increase the innate immune system. Okay, so they are going to increase the macrophages, the natural killers. They are going to actually, right, so I'm gonna, here's a star. T helper cells are also going to increase the co-stimulation of the cytotoxic T cells, so essentially ramping up how many of these are actually activated. Let's see, it is also going to lead to IgG antibodies, all of which are going to be particularly good at attacking intracellular substances, right? So this particular thing is intracellular, and so we're going to ramp up all of the things that are great at dealing with intracellular stuff, including cytotoxic T cells, right, which are also going to be attacking and physically killing, right? These guys never kill, they're just helping to ramp up the process. But cytotoxic cells can actually kill intracellular pathogens, right? Okay, so what is the other option here? All right, so again, T helper 1 enhances the innate immune response. And another detail here, IgG really helps with intracellular pathogens because it induces complement, yet another one of these innate immune responses. Okay. The other option here is inducing the differentiation into T helper 2 cells. T helper 2 will be triggered by the presentation of a T helper 0 cell with an extracellular pathogen, right? So something that's just kind of floating around. Maybe this would be a piece of a tapeworm, right? Maybe a piece of an E. coli bacteria, something outside of our cells, as opposed to a virus, which gets into our cells and kind of hijacks its own machinery. Okay, once again, these extracellular pathogens are presented on class 2 MHC complexes, but because it's extracellular, it's going to trigger the T helper 0 cell to initiate a slightly different response. That response is to activate particular components of the immune system, such as eosinophils, basophils, and mast cells. Remember that these guys are granulocytes. These granules are filled with different inflammatory compounds. And so that is very fitting for extracellular pathogens. Okay, also, T helper 2 cells are going to activate our B lymphocytes in a very large way, so even more so than the T helper 1. And so we're going to get into that a little bit in our next lesson together, but essentially, B lymphocytes absolutely need activation by T helper cells. That's where the co-stimulation comes in. T helper cells need to activate B lymphocytes. And so without T helper cells, such as in the case of HIV, right? HIV hijacks T helper cells so that they cannot activate all of these things that we're talking about, most importantly, the B lymphocytes. And so that's why the immune system of HIV patients and then later AIDS patients, right, immunodeficient patients is so compromised, right? We need the T helper cells to activate the rest. Okay. And so essentially the T helper 2 cells in particular are going to enhance the humoral response, right? So the B lymphocytes. So again, let's add this up here to our diagram. Once again, we're going to have an APC, which has an MHC2, actually here in red, and I'm drawing this in red because the MHC2 is recognized by the CD4, MHC1 is recognized by the CD8, and so here is MHC2. Okay. And again, we're going to present a particular extracellular pathogen, so outside the cells, a tapeworm, whatever, and we simultaneously have to co-stimulate the T helper 0 cells. Okay, so here is co-stimulation. Okay. With the concurrent presentation and co-stimulation, this T helper 0 cell is going to be activated. Right? Hopefully this is starting to seem familiar now, and it is going to proliferate. We don't want just one single cell, we want tons of these particular T cells. These are the T helper 2 cells. All right, so what do? They essentially increase the humoral response. All right, so increase the B cells. All right, and in our next lesson together, I will show you exactly how these T helper 2 cells are going to activate those B cells as well. So there'll be another half of the diagram over here. Okay, but for now, let's just add a couple more details here. These can also increase things like eosinophils, basophils, and mast cells. Right? If you remember from our discussion of the cardiovascular system, these guys increase allergies, right? Also, the T helper 2 cells can trigger the B cells to release IgE antibodies. And as I said, different antibodies are responsible for different types of activities at different locations. These are the ones that are associated with allergies. All right, so the thought here is that T helper 2 is trying to increase a response against extracellular pathogens. Right? If you are inhaling a lot of pollen and stuff, and for whatever reason, your T helper cells or your APCs and T helper cells together are determining that that pollen is not good, we need to get rid of it, then we are going to launch a response that produces a lot of extra inflammation and allergy symptoms, including with these IgEs. Right? And so the other option, of course, is recognizing the pollen as not being a threat. And so there won't be this presentation and co-stimulation. And so all of this would never happen. Right? So it's just a determination by your body, your immune system, with this process that determines pollen as the extracellular substance, right? It's not breaking into your cells or anything, right? We're not going to kill it with a cytotoxic T cell, but if this extracellular foreign substance is determined by these cells to be dangerous, we are going to launch a stronger humoral response and also lots of extra inflammation, as well as mucus production and all of that kind of lovely allergy stuff. Okay. And so that leads me to the final point that I want to make in this presentation here, and that is that in a normal healthy individual, there's actually a balance between the T helper 1 responses and the T helper 2 responses. And so this, right, it's important to have both, right? If you have a tapeworm, you really need to deal with that, right? So your T helper 2 response is going to help you to eliminate that. But then on the other hand, if there isn't this nice balance between the two, then you might overreact to certain types of extracellular substances. And so this balance between 1 and 2 can be impaired for various reasons, or it can be overactive if this balance is lost. If for a variety of reasons, this shifts more to T helper 2 as opposed to T helper 1. Okay. T helper cells can also release inhibitory cytokines so that only one type is dominant during a particular response. And again, this is your body determining that, you know, pollen is really scary, right? We don't want this in our body. And so we are going to launch or we are going to dominate the response to pollen. Anytime we see pollen in our body, the T helper 2 cells are going to deal with that, right? So that is when you would develop allergies, as opposed to your body deciding, nah, it's kind of okay, like we need to get rid of it, absolutely, but not launch a particularly strong response. Okay. So there's a lot of really cool readings about this, and I really want to simplify this, as hard as that may be to believe, because it's just so crazy. This is simplified, but I highly encourage you to read more about this, especially if you have allergies, especially if you are really interested in the immune system, because our immune system is just so very important, particularly in the age of COVID. So that is all I want to discuss for today. one final portion of the immune system is going to be presented in our next lesson together. Thank you.